Okay, so now we're pretty familiar with Mendel's classic laws of segregation and independent assortment. But as it turns out, genetics in the real world is a lot more complicated. So let's start with incomplete dominance. So in this pattern, the heterozygote has an intermediate phenotype. So it's not dominant and it's not recessive, but it's somewhere in the middle. So a classic example is found in certain plants. When you cross a red flowered plant with a white flowered plant, the result isn't red or white, it's actually pink. When you cross two of those pink flower plants, you can actually get offspring in a 1-to-1 ratio. So 1 red, 2 pink, and 1 white. So the alleles are still distinct, just neither one fully dominates the other. There's also a human example. So familial hypocholesterolemia is a disorder where the incomplete dominance affects cholesterol metabolism. So people with one mutated allele have high cholesterol, and those with two mutated alleles have extremely high cholesterol and would be at serious risk for early heart disease. So here's an example here of the red and the white flower. And when you cross them, you can get in the F1 generation all pink. If you take that F1 generation, which is all pink, and you cross it, then you get this in the 1-to-2-to-1 ratio. So just as a reminder, what we were talking about previously with Mendel was complete dominance. So it was either going to be green or yellow pea pods. When we talk about incomplete dominance, one of the phenotypes is blended. So if we're talking about flower color, we have red and white, and our intermediate phenotype or the blended phenotype would be pink. We also have codominance, which there's not a separate slide on and it's not discussed separately in your textbook, but it is a component of what we're going to talk about next, which is ABO blood typing. So if we're talking about codominance, that means both alleles are going to be expressed. So I just want to review this with flower color. So in complete dominance, let's stick with red and white flowers. Actually, I'm going to switch to cows because I think it's easier to conceptualize. So let's do cow color. If we have brown and white cow and their offspring are only brown or white, that's complete dominance. One allele is dominant over the other. If we're talking about incomplete dominance, we can get a blending. So we can have our possible phenotypes be brown or white. The heterozygote would be blended, so like a light tan, something like that. Now, in codominance, both alleles are expressed. So if we have a brown and a white cow, we would have a brown and white spotted cow. So both expressed at the same time. So that's the difference between complete, incomplete, and codominance. Because now we have to talk a little bit about traits with more than two alleles. So that's our ABO blood type system. And this has both a combination of complete dominance, which is why you see a capital letter and a lowercase letter, as well as codominance, where we can have our heterozygote represented like this here. So in incomplete dominance and in codominance, we write the genotype a little bit differently. And for blood typing, we always use the letter I. So I with the superscript representing what allele it has. So when we're talking about blood typing, we're talking about three alleles being involved. We have capital I with superscript A. That's going to code for your A-type antigens on the surface of your red blood cell. So if you have blood type A, you have the A antigen. If you have the capital I with a superscript B, that codes for B-type antigens on the surface of your red blood cell. If you have a lowercase I, that means you do not have any A or B antigens on the surface of your cell. And if you have blood type with both an A allele and a B allele, then you have both the A and the B antigen on the surface of your red blood cells. So these alleles can combine in different ways to create our four main blood types, which again are A, B, AB, and O. Something to think about, this is probably a little bit too much information, But something to think about is that blood type AB is considered the universal recipient because there are no antibodies against any antigens in that person's serum. And blood type O, that's considered the universal donor because it doesn't have any antigens on the surface of its red blood cell. So if a patient had any antibodies in their serum at all, it wouldn't matter because there's no antigens for those antibodies to attack on somebody with blood type O. So like I mentioned, the system also demonstrates the codominance. If we're talking about blood type AB, both alleles are being fully expressed. They're not being blended. It is not an intermediate phenotype at all. So this is an example of the ABO blood typing here. So some traits are too complex to be governed by a single gene, and that's where polygenic inheritance comes in. And these traits are influenced by 2 or more genes, often on different chromosomes. So each dominant allele is going to add a dose of an effect, so the result is often a continuum rather than a clear-cut category. So think of traits like skin color, height, and eye color. They don't fall into a few boxes, but rather they form a bell-shaped curve across populations, which you can see over here. We don't just have really short people, really tall people, and intermediate people. We see this kind of continuum of height. Same thing with skin color. And things can get even more complicated when you factor in the environment. So traits like cleft lip, diabetes, allergies, schizophrenia, and even some cancers are multifactorial, meaning they're influenced by both genes and environmental factors. So for example, you might inherit a genetic predisposition to diabetes, but your diet, your lifestyle, and exercise habits will play a big role in whether or not you actually develop it. So we can also talk about how the environment can literally change gene expression. So take skin color as your example. Your genes determine how much melanin you can produce, but UV exposure triggers how much melanin your skin actually does produce. So that's why people tan and why populations closer to the equator tend to have darker skin. More UV light equals more melanin production for protection. So even if your genotype isn't fixed, your phenotype isn't always set in stone. So environmental signals can modify how your genes are going to be expressed. Okay, so let's talk about how genes interact with each other, especially for traits like eye color. It's not just one gene that's turning eyes blue or green. Instead, there's multiple genes that influence different pigments. So these pigments can mix in various ways, giving rise to a spectrum of eye colors. So that's why predicting eye color isn't always straightforward, even if you know the parent's traits.